first review the electrochemical equilibrium. So I've got your 5 players up here. All right. What are they in abundance inside and outside the cell? So I want you guys to take a minute, since this is now, so we talked about it, we talked about cells, we talked about it with muscles. This is the 3rd time we brought up this principle. So see what you remember, what's in greater concentration, how are you going to draw it inside and out the cell. You can talk to your neighbor about it too. All right, so I would start with our proteins, which are going to be in greater concentration, which side? Inside the cell, right? And this is really what, and they are negatively charged. This is really what allows the equilibrium to be set up. All right, and then what else is in greater concentration within the cell? Potassium, good. Okay, so there's not as much potassium out here, and there aren't as many proteins floating around here. So in relative concentration, most of them are inside the cell. Well, that makes it easier for the rest because they're just the opposite, right? So we have a lot of sodium out here, not as much in here. A lot of calcium out here, not as much in here. And chlorine. And there are leaky channels, and there are sodium-potassium pumps, which are maintaining this difference between positive and negative overall charge. And if there's a difference, that's called a potential, okay?
Wait, sorry, can you spell that more time out here?
Yep. If there's a difference in charge inside and outside of the cell, that's what we call our potential. And that helps maintain our voltage. Yes?
I have a question. So if proteins are too big to move outside of the cell
Mhm.
and they stay inside of the cell, are the proteins that you do different proteins?
Yes. Yeah. Lots of different types of proteins. But there are some proteins that float around in the blood and the interstitial fluid. All right. And we've got 3 different types of, well, we've got lots of different types of channels, but 3 different channels that can change the potential, change the difference, which can be stimulated under different conditions. So the first type we're going to talk about are, so if we drew a gated channel here, this is a protein that's embedded in the membrane. Okay, and it has this little gate over it. And well, I'll just spell it out. Our first type is the voltage regulated gated channels. And so what happens here is if there is an electrical stimulus, this is clearly a lightning bolt, okay, it will stimulate it to open. And then ions would flow in or outside of the cell depending on what type it is. All right, and then we had, so we're familiar with these. We're also familiar with the second kind, which opens when
The channels are different based off how they open, or
what stimulates them to open?
okay, so the first one
Voltage, an electrical signal. Yeah. All right, we also have chemical regulated ones, all right? So I'm going to draw another one over here. This one has a little spot there, and my swinging gate. It's horrible. All right, and what was the chemical gated or chemical regulated gated channel which we had with at the neuromuscular junction? Yeah, the acetylcholine receptor. All right, so you have some chemical which binds here, and the binding of that chemical causes it to open. And generally, this is a neurotransmitter. With our neurons, it is a neurotransmitter. At the neuromuscular junction, it was a neurotransmitter, acetylcholine. Okay, and our third type, which we talked about in the quiz, is mechanical, right? Where some sort of physical deformation or pressure on the cell membrane will cause it to open. So I'm going to draw a finger here. Thank you. Using the power of touch. It deforms the membrane, and then that causes it to open. Ions flow in or out of the cell. All right, but this is all, the only thing new we have here is the mechanical gated, or the mechanical regulated, which is going to be important for our touch, pain, pressure receptors. All right, so now let's move on to the neuron and an action potential. All right, one thing to note, an action potential is an all or nothing event. So you can't have a half of an action potential. And once it goes to completion. It can't stop in the middle and go backwards. Okay? And so the action potential is the initiation and propagation of this electrical signal, or this change in voltage down the cell. And there's 4 stages. The first stage is just at rest. Which we've determined before, at least for these types of cells, is at negative 70 millivolts. So the difference between the ions inside and outside the cell is at negative 70 millivolts. This is due to the leaky channels and the sodium-potassium pumps setting up that difference. So this is stage 1. What happens is you have some sort of stimulation of those 3 types, mechanical, voltage, or chemical, which causes ions to flow into the cell, positively charged ions, which is going to make the cell less negative. And if it goes above a certain point, which we will call the threshold stimulus, you enter the second stage of the action potential called depolarization. Yes?
So is the portion underneath the threshold stimulus that's rising, is that still part of the resting potential?
No.
What is it part of?
So the resting potential, we'll talk about it in a minute, actually, when we talk about graded potentials. But this essentially is depolarization caused by the stimulus. Okay, and once the stimulus is strong enough and it passes the threshold, then we're in the action, the depolarization irreversible stage. Okay, but we'll talk more about it in a second. So, what's going on at the neuron? Okay, during step 2. Here we have a sodium channel. And during depolarization, it opens. And sodium is going to flow from down its concentration gradient into the cell. Which, because it's a positive ion, is going to make the potential less negative. To the point where it's going to flip from being a negative potential to being a positive potential, all the way up to 30 millivolts. Yeah.
So is the sodium channel like voltage regulated?
It could be voltage regulated, it could be mechanical, it could be.
Okay, so it'd be any kind of stimulus that opens up that channel.
Yes.
And that creates the stimulus.
Yeah. So we aren't specifying which type, it could be any type depending on the neuron. Yeah.
Well, didn't we just talk about like that one open and will reach threshold?
So, well, some of them will open, depending on the stimulus. Again, we'll talk about it in a second, about the threshold. But essentially, this amount of sodium has to enter the cell past the, if it could go like this and go back down if it doesn't go past the threshold. So some amount of sodium can go in. Yeah.
Because once all the voltage regulated sodium channels open,
Then going all the way, yeah. But yeah, we'll talk about that in a second. Okay, at this peak point, okay, what happens then is you get a repolarization. Okay, and well, you can see that we are going back to this negative potential, negative voltage. And what's happening here then at step 3 are two things. One, the sodium channel is deactivated, which is slightly different than closing. Just has another piece that prevents sodium from getting in there anymore. So we'll put a 3 next to this guy.
So the
second step, it opens. Third step, it's deactivated, so it blocks sodium from coming in. In addition to that, our potassium channel opens. And now, because we have lots of potassium inside the cell, it can flow outside of the cell.
And what kind of channel is the potassium channel?
It's a voltage-regulated potassium channel. Because it is regulated by that, it just kicks in after the sodium
So it's a
kicks in.
voltage-regulated potassium channel?
Yes. So that's why we have a decrease in our potential back down to 70. Well, what happens after that is it goes down even further. So we call this hyperpolarization. And essentially more sodium goes, or more potassium goes out of the cell than sodium came into the cell. So now we're even lower than negative 70. And after that, then the action potential is complete. And we return to the resting potential. Okay, the resting potential is maintained by the sodium-potassium pumps. All right, so you got to get all that sodium out of there, and you got to get all that potassium back in there. So you're going to use sodium-potassium pumps to do that.
Why are you not saying no restoration is a fifth step?
We can. We'll put that on there. And then you're at the resting potential again. All right, well, this action potential, like we said, is an all or nothing event. And if we were to give these, let's give these different colors. So let's say the resting potential is red, depolarization is blue, and repolarization is orange, and hyperpolarization is green. So at rest, this whole part of the neuron is in a resting state. But once our cell body and then end to the axon hillock has been stimulated, it's going to go through the different stages. So first you're going to get depolarization here. And then that will go on to the next section. You'll get depolarization further down. But behind it, you'll have repolarization. And then that will continue as the signal goes down the axon. All right, so you're going from resting to depolarization. from depolarization to hyperpolarization. Sorry, repolarization. And then after that, you get hyperpolarization. And it's just going to go in a sequence of events down the axon. And then the resting potential will be restored behind it. And what that allows it to do is it allows it to go in the same direction. So if you were to depolarize and then repolarize, then you have a potential. Let's say, so if I came down here in the middle and I depolarized, what did I use? Blue? And I depolarized right here. And then it would repolarize behind it. The repolarization and the resting potential are the same. They're both at -70. So if they were both the same, this signal could go in both directions. Okay, but because we have hyperpolarization on one end, that gives us directionality, so the signal can only go in one direction and continues in that direction. So that's why it overshoots. So we can't go backwards. We can only go one direction. Yeah?
Monday we talked about unipolar and dipolar, and I asked about that, where the axon hillock is and how that works.
Yeah, I don't fully understand. But they do have dendrites on one side and axon terminals on the other. So I imagine it just doesn't start with the axon body or in the cell body.
The similar thing has to happen in the dendrites in order to even propagate to the cell body.
Yeah, so maybe it doesn't, maybe the cell body isn't involved, is what I think. I'd have to look it up though. It's a good question. Yes?
So basically the cycle keeps going and the only thing that keeps it going is hyperpolarization.
Hyperpolarization is the only thing that keeps it from going backwards. But because it's depolarized, you essentially have these same voltage regulated channels all the way down the axon. So as the voltage changes on the left, it's going to stimulate it to the right and continually stimulate it. So the sodium channels will open, and the potassium channels will open.
So it's just kind of a give and take, like, and then go like that.
Yep. Yeah, so it's going up and down, overshooting up and down, and all the way down. And so that's how an action potential works then. Until it gets all the way to the end of the axon terminal, and then the next thing happens. You have a neurotransmitter there, transmits to the next signal or whatever. Yeah?
Can you give us an example of how you test this on the exam?
Sure, yeah, you could do this graph, and I could ask you what's happening at each step with the different channels. I could have you demonstrate how hyperpolarization prevents the signal from going backwards. Lots of good questions on this, yeah.
Is the worksheet, is it going to have some figures for us?
Yeah, worksheet has some good figures on it too. All right, so I find this complicated, abstract. If you're only looking at the graph, you're like, all right, well, what does that mean? But when you look at it in relation to what's going on in the neuron, what you know about the electrochemical equilibrium, what you know about the different channels and the influx and outflux or efflux of the different ions, then it all makes sense, right? Or it will for the test. Okay, so I said you guys were kind of jumping ahead about our graded potentials. Graded means All right, they can go up or down or add or they are not all or nothing. Okay? They are what we call additive. All right, and these all occur below the threshold. Because if anything goes above the threshold, what happens? Our action potential, right? It'll go all the way through those steps. Okay, so there's two types. We have the excitatory post-synaptic potential and the inhibitory post-synaptic potential. And the same mechanisms are in play where you have sodium channels Yeah, this is the figure from the book. All right? So what I want you to do is, we've got a number of things here. We have both the graded and action potential. Can you label what's on here? Yes, you can. So take a minute and see what you can label on this graph.
You really had to do the exact opposite colors, didn't you?
I don't know, I just randomly pick colors. Yeah, so see what you remember, see what you can label from this. Not from the book, from what you remember. Right? You can go to the book next. See what we can come up with. Well, we already have the EPSPs and the IPSPs labeled for you, right? So that made it a little bit easier. But essentially you have what's going on with the EPSPs. The potassium channel's open. And for the IPSPs, sorry, I said sodium. I wrote sodium and said potassium. And potassium channel's open. Yeah.
I think for the IPSPs, it helped me to remember that it can also be chlorine, which is negative.
Yeah. So yeah, we're gonna keep it simple for this. Yeah.
Okay.
But yeah, any ion that reduces it, makes it more negative would also work. All right. What do we have at this point here? Okay, good. Resting potential. I heard somebody over there say it. All right, and the resting potential is being maintained by what process? All right, sodium-potassium pumps, which requires ATP. So this is part of the cost of being alive, is just maintaining the electrical potential across your neurons, constantly being used. All right, so we have some sodium channels opening, and you would say, what's going on here, repolarization or depolarization? All right, depolarization. And at the IPSPs, repolarization. All right, and then we have a short period here where it's yellow, where it's decreasing. What's going on there? Right, yeah, so we're sodium potassium pumps are starting to restore it, but then we get another EPSP, right? All right, how about these two together? What do we got going on? Right, so they were back to back. So we get some temporal summation there. And this one, I think it's harder to show, but you have a greater increase over about the same amount of time. So we can assume this is spatial summation. Where two of them occurred at different points at the same time. So like this one and this one. And then what happened? An action potential, right? Because we went above the threshold. So at this point we have depolarization of the action potential, repolarization of the action potential, hyperpolarization, and restoration. There's a lot on there, right? Okay, but this is a great test question. Because it asks you to like understand all the things at once, right? It has the axon on there. Okay. Or I could say, or I could number these, and I could say, show what happens if 1, 2, and 3 fire at the same time. You show me.
You should.
Yeah.
How is that green one in the middle there? How is that spatial summation?
Well, we're just assuming that it is a bigger jump in the same amount of time. And so we're using our imaginations a little bit.
Okay.
But we're saying 1 and 2 fired at the same time, and so you have, rather than this little increase, we have this big increase.
I thought spatial summation made it kind of state neutral, state static.
Oh, that would only be if an IPSP and an EPSP fired at the same time. Right? So if 2 EPSPs fired at the same time in different locations, they would add on to each other. It happened at different spaces. If you had 2, if they're at the same time, occurring at different locations, at different spaces, that's spatial summation. If you have 2 occurring one right after another, that's time, that's temporal. So the example I gave in before was spatial summation of an EPSP and an IPSP. Right.
But if they're both EPSP, then it's both going to go up.
Yes, you got it. So this is spatial summation of 2 EPSPs, right? Yes. No, these are both excitatory. It's only inhibitory if it's going to go down. So all these pink ones are inhibitory. Is that clear? Not clear. Okay, but it's okay. Terminal though, this should be easy, right? Because this is pretty much the same thing as the neuromuscular junction, except we've got 2 neurons instead of a neuron and a muscle. So what we have is a presynaptic neuron. And then on the other side, you have a postsynaptic neuron. And this is called what? The synaptic cleft. So if it's before the synaptic cleft, it's presynaptic. If it's after, it's postsynaptic. Everything else is the same. Well, almost everything else. You have vesicles filled with, well, we're just going to say neurotransmitter, but it could be acetylcholine.
Yeah, we should probably make this down somewhere.
All right, and when an action potential comes down the axon terminal, these, it will stimulate exocytosis, right? Yep.
Is it still a voltage regulated channel that opens and allows calcium ions in?
Yeah, it's not necessarily calcium. Actually, with the neuron, it's going to be sodium.
Okay.
But yes, a voltage regulated sodium channel. And then on the other end, when the neurotransmitter binds, the channel opens. And you have a repolarization event, which leads to another action potential, or if it's not strong enough, just fades away. All right, but because you guys just got tested on this, this is nice and fresh. Just a few minor differences.
So the minor differences are just that we're not calling it acetylcholine, we're just calling it neurotransmitter.
Right, because it could be any number of neurotransmitters depending on the neuron, what it's exciting. And it could have multiple neurotransmitters
And then
depending on what it's exciting.
Sodium coming in through that other channel up there.
Yep, sodium instead of calcium. And then you have a presynaptic neuron and a postsynaptic neuron rather than the neuron and the muscle cell. Okay, so the last thing we're going to do is split up the nervous system into its different parts, okay? All right, yeah, it's nothing to be nervous about. All right, so the first split we've already talked about. The central nervous system and the peripheral nervous system. The central nervous system is made of your brain and spinal cord. And the peripheral nervous system is made of your spinal nerves, and cranial nerves. Let's just say nerves.
But on the test, you'll want us to know the specific nerves, right?
Well, we'll get into more detail when we talk about the brain and stuff, but at this point, just
The central nervous system are the nerves of the brain and spinal cord, and then peripheral, just the nerves of.
no. So within the brain and the spinal cord are the central nervous system. There are nerves that go into the brain, and there are nerves that go into the spinal cord. Those are peripheral. So we'll just say the nerves. That's even better. 2 from that actually is the next division. You have afferent nerves and efferent nerves. Okay, afferent is sensory. Efferent is motor. So from your motor neurons, then, you have somatic, which are voluntary, right, under conscious thought. And autonomic, which sounds a lot like automatic. They are involuntary. One more split, okay? Our autonomic then splits our involuntary into the joy nerves. I'm just kidding. Sympathetic and parasympathetic. Sympathetic is our fight or flight response. It's really more broad than that, but. And then parasympathetic is rest and digest. Okay, and then the parasympathetic, I'm just kidding, that's it. Seems like you could go probably on forever. That's it. So at every point, though, you have a dead end, and then you have another branch. So you got 4, 1, 2, 3, 4, 4 branches, 4 dead ends. Easy. All right, so afferent, just to kind of add a little detail, right? So sensory information is going to go up into, up into the peripheral nervous system and then into the central nervous system. Efferent is going to be coming away from that, okay, in response to your sensory information. All right, that's all we'll add for now. But we'll refer back to this because we're going to go through all these different steps back and forth.